Hello everyone, welcome back. Today I am here with this important topic, that is microsporogenesis. It means that formation of microspore from diploid microspore mother cell inside the microsporangium. So pollen sac के अंदर कैसे microspore mother cell से microspores बनते हैं और pollen grains बनते हैं, that process is known as microsporogenesis. को हम डिटेल में पढ़ेंगे in this lecture. Microsporogenesis, it is a process of formation of microspore from diploid microspore mother cell. So during this process, this sporogenous tissue in microsporangium, which is diploid, act as microspore mother cell either directly, or it can undergo several mitotic division before acting as microspore mother cell. Then this microspore mother cell undergoes the process of meiosis, which results in formation of microspore tetrad. And finally, जो microspore tetrad है वो वहाँ से pollen grain separate होते हैं because they have individual callase layer जो हम last lecture में discuss किया था, results in formation of pollen grain, which is a haploid structure. So formation of microspore inside the microsporangia is known as microsporogenesis. A diploid cell in microsporangium that acts as microspore mother cell, that is MMC, also known as microsporocyte or pollen mother cell. It undergoes a process of meiosis to form haploid microspore. Microspore development is similar up to Meiosis-I. जो Meiosis-I है वहाँ तक सभी तरह के microspore की development similar है. After that, it can follow one of the two patterns. The first pattern is, in first pattern, each nuclear division may be followed by cytokinesis, means immediately followed by wall formation. In the successive type, or the wall formation occurs simultaneously only at the end of Meiosis-II. जब Meiosis-II का end हो जाएगा तब ही इसमें walls बनेगी, that is simultaneous type. That we'll discuss in detail next slide. This determines the arrangement of pollen in tetrad. So जो pollen tetrad बनता है उसकी arrangement कैसी होगी ये determine करता है ये जो divisions हैं, that is successive type or either simultaneous type. So this is microsporogenesis. It involves the formation of this microspores from this diploid sporogenous cell or microcyte. So first division is successive type, as we have discussed earlier. In successive type, karyokinesis is immediately followed by cytokinesis, that is division of cytoplasm along with formation of cell wall. So जैसे ही nucleus का division होता है वैसे ही wall formation होती है. So each nuclear division is followed by cell wall formation, that is cytokinesis, thus forming a dyad or tetrad that we can see in this animated photograph. The cell plates are laid down in centrifugal manner. जो cell plate formation है, that is in centrifugal manner, and examples are Canna, Lilium, Mayaca, Senseviera and Zia. तो इन plants में इस तरह की division होती, that is successive type, in which nuclear division is immediately followed by cytokinesis. Second type is simultaneous type. इसमें जो cytokinesis होता है, that is at the end of Meiosis-II. So in this photograph we can see here simultaneous type. ये dicots में होता है और successive type mostly monocot में होता है. First nuclear division is not followed by cytokinesis. इसमें immediately cytokinesis नहीं होता है, but rather the wall formation takes place simultaneously at the end of meiotic division. जैसे ही meiosis division end होता है वैसे ही इसमें wall formation होती है at the end of Meiosis-II, that we can see in this photograph. The cell plates are laid down in centripetal manner means towards center की तरफ को जो cell wall है बनती है. And examples are Electra, Helleborus, Nicotiana, Magnolia and Melothera. So ये इसके examples हैं जिनमें ये होता है. So simultaneous type occurs in dicot while successive type occurs in monocot. Then we have 5 different types of microspore tetrads which are reported to occur in angiosperms. Angiosperm में 5 तरह के microspore tetrads पाए जाते हैं. Firstly, we have tetrahedral. As the name indicate, इसमें जो microspores हैं, 3 microspore visible होंगे फ्रंट साइड में और एक होगा इसके पीछे. So this arrangement is formed as a result of simultaneous division. So जो simultaneous division है उसके result से tetrahedral पैटर्न बनता है pollen tetrad का. where 4 microspore lies at 4 corners of the tetrahedral. Only 3 microspores can be seen in front view. तो front में हमें सिर्फ 3 ही microspore दिखते हैं, and the fourth one is located on the back side, और चौथा है इसके पीछे. It is most common type of tetrad arrangement. तो ये बहुत ही common तरह का tetrad arrangement है in angiosperms. Examples are alerta, then we have Helleborus, Nicotiana, Magnolia, and Meiosis-I. Second type of tetrad is isobilateral. It is again one of the commonest tetrad. In this arrangement is formed as a result of successive division. ये बनता है successive division के result से, where 4 microspore lie in one plane and are visible. So ये जो microspores हैं ये चारों ही एक ही plane पे होते हैं, तो हमें ये front side से चारों ही pollen grains दिखते हैं. It is very common type again, और ये पाया जाता है कैना में, लिलियम में, मैका और सेंसिवेरिया और जिया में. Next comes Decussate arrangement of pollen tetrad. The microspores are arranged in 2 pairs, where each pair is at right angle to the other. So ये जो Decussate arrangement है, इसमें जो microspore 2 microspore हैं, इनके right angle में present होंगे next to. So एक इसके पीछे present होगा. So that type of arrangement is known as Decussate arrangement, और ये पाया जाता है Artiplex में, Crocus में और Magnolia में. Next comes T-shaped pollen tetrad. As the name indicates, it looks like a T. So the microspore are arranged in a T-shaped manner. Examples are Aristolochia and Butomopsis. तो इसमें जो पोलन टेट्राड्स हैं, दो पोलन ग्रेन्स होंगे ऊपर की तरफ, और इसके नीचे सिंगल पोलन ग्रेन प्रेजेंट होते हैं, which gives an appearance of T. Next is linear. This is one of the simplest type. इसमें जो pollen grains है, they are arranged in linear fashion. So microspores are arranged in linear fashion. For example, we have Halophila. Then जो tetrahedral और isobilateral type है pollen tetrad के, they are the most frequent, most common types of pollen tetrad which are found in angiosperms. Then Aristolochia elegans. This is important fact. जो Aristolochia elegans है, या हमारे पास है Spargenium erectum, ये जो 2 plants हैं, इनमें सभी तरह के means 5 types of pollen tetrad are found in the same anther. तो एक ही anther में सभी 5 तरह के pollen tetrad पाए जाते हैं. Sometimes pollen grains fail to separate at maturity. So pollen grains separate नहीं हो पाते हैं, they are carried as a unit. तो single unit है, उन्हें carry करते हैं during pollination, as in case of Philodendron lanuginosum. So एक plant है Philodendron lanuginosum, इसमें जो pollen grains है वो separate नहीं होते हैं, वो as a single unit ही pollinate होते हैं. Pollen tetrad in a pollen sac remain together to form compound pollen grains in some plants. So जो pollen tetrad है वो separate नहीं होते हैं, वो ऐसे ही रहते हैं साथ में, and they behave as a compound pollen, as in some plants like we have Acacia, Anoa, Drosera, Dremis, Eloida, and Typha. So in plants में as a unit, single unit, सभी pollen grains साथ में ही pollinate होते हैं. We have some families like Asclepiadaceae, which is commonly known as milkweed family. इसमें एग्जाम्पल आता है Calotropis. Then we have Orchidaceae. इनमें जो pollen grains होते हैं, they are present in a single pollen sac and are agglutinated to form a highly specialized structure जिसे कहते हैं pollinium. So pollen का एक composite structure present होता है, जो कि as a whole unit, as a single unit transfer होता है, pollinate होता है. That structure is known as pollinium, which is a composite structure. So ये जो 2 sac like structure है, इसके अंदर present होते हैं pollen grains जो एक single unit की तरह behave करते हैं, and this is joined with the help of this disc, and these arm like structure are known as retinaculum. और ये है Calotropis का pollinia, जो as a single unit behave करता है, और as it is ही ये transfer होते हैं, pollen grains separate नहीं हो पाते. Then in some members of Orchidaceae, like we have Habenaria, Hominium, Orchis, pollen grains remain loosely arranged in a small packets. जो pollen grains हैं वो loosely arranged रहते हैं small packets की form में, जिन्हें कहते हैं mesulae. So these structures are known as mesulae, which are found in some orchids. Occasionally the microspore tetrad may undergo further division to produce polyars. So कई बार दैट इज़ एन एनोमैली कंडीशन, एनोमैली है, एबनॉर्मैलिटी है, जिसमें माइक्रोस्पोर टेट्राड फिर से डिवाइड करते हैं बाय मेयोटिक डिवीजन टू फॉर्म पॉलीआर्स. द नंबर ऑफ माइक्रोस्पोर इन ईच ग्रुप कैन बिकम 8 भी हो सकते हैं, लाइक वी हैव हाइफने, then 11 in Cuscuta and even 22 in Thunbergia Meiosis-I. तो इनमें प्रेजेंट होते हैं डिफरेंट नंबर्स ऑफ माइक्रोस्पोर्स. दिस फिनोमेनन इज नॉन एज पॉलीस्पोरी, इसे कहते हैं पॉलीस्पोरी, और ये होती है एज अ रिजल्ट ऑफ माइटोटिक डिवीजन विच अकर्स आफ्टर द फॉर्मेशन ऑफ पोलन टेट्राड. जब पोलन टेट्राड बन जाता है उसके बाद फिर से पोलन ग्रेन्स दे अंडरगो द प्रोसेस ऑफ माइटोसिस विच रिजल्ट्स इन दिस abnormal phenomenon which is known as polyspory. So it is brought about by additional mitotic division of the constituent microspores in tetrad, and this phenomenon is considered as an anomaly. This is an abnormality. So these are the structures which this is the same images which shows the maculae, which are the loosely arranged pollen grains जो कि एक सिंगल यूनिट की तरह बिहेव करते हैं. These structures are not. Next comes ANTHER dehiscence. It is the phenomenon of opening of anther to release pollen grain. This is known as ANTHER dehiscence. जब anther mature हो जाते हैं और उसके अंदर pollen grain बनते हैं तो anther they will undergo the process of dehiscence, which means उसमें एक opening create होगी जिसके through pollen grains बाहर release होंगे. So that process is known as ANTHER dehiscence. So release of pollen grains from this mature anther is known as ANTHER dehiscence. दिस में भी लॉन्गिट्यूडनल, जो एंथर डेहिस्सेंस है वो लॉन्गिट्यूडनल हो सकती है, ट्रांसवर्स हो सकती है. दिस इज लॉन्गिट्यूडनल मीन्स यहाँ से ये जो एंथर है वो इसमें स्मॉल ओपनिंग बनेगी जिससे पोलन ग्रेन्स बाहर निकलेंगे. देन इट में भी ट्रांसवर्स हो सकती है, यहाँ पे ओपनिंग स्लिट्स बनेंगे इसमें ट्रांसवर्सली फ्रॉम विच पोलन ग्रेन्स कम्स आउट ऑफ द एंथर. इट में भी वेल्वुलर, इट मीन्स इट हैव वेल्व्स, बाहर से वेल्व्स की तरह स्ट्रक्चर होंगे जो ओपन हो जाते हैं जिससे पोलन ग्रेन्स बाहर आ जाएंगे. देन इट मे बी पोरस, इट मीन्स इट हैव पोर्स एट इट्स टिप जिससे पोलन ग्रेन्स बाहर निकलते हैं. एंड इरेगुलर इन फ्लावरिंग प्लांट. सो देयर आर डिफरेंट टाइप्स ऑफ dehiscence pattern which are followed by anthers. Anthers already have predetermined lines of dehiscence which are indicated by thin walled cells. तो जो एंथर्स हैं उनमें पहले से ही लाइन ऑफ dehiscence पहले से ही प्रेजेंट होती है जो बनी होती है थिन वॉल्ड सेल से. एंड दिस एंथर dehiscence इज अ वेरी कॉम्प्लेक्स फिनोमेनन, कॉम्प्लेक्स प्रोसेस है ये, रिक्वायरिंग सेलुलर डिफरेंशिएशन एंड डिहाइड्रेशन ऑफ सेल. Then cellular differentiation is in the form of lignin thickening on the inner and radial walls of endothelium, which plays an important role in anther dehiscence that we have already discussed. जो endothelium के ऊपर special thickening bands होते हैं, U-shaped bands, they show hygroscopic movement और वो important role play करते हैं anther dehiscence में. The mechanical layer also, that is fibrous or contractile layer which surrounds the loculi except at the stomium. So the mechanical layer भी इसमें present होती है जो surround करती है loculi को, anther sacs को, that except at stomium. Stomium के region पे जहाँ पे anther की two structures are responsible for anther dehiscence. One is the stomium and second one is the mechanical layer, that is endothelium. ये दो structure responsible है anther dehiscence के लिए. Both stomium and mechanical layer is responsible for dehiscence of anther. This is followed by disintegration of sterile strip which is present between the two pollen sac at each lobe. So जो दो pollen sac हैं इनके बीच में जो sterile cell की strip है वो भी disintegrate हो जाती है. This leads to formation of a single cavity in each pole, जिससे कि एक पोल पे एक single cavity बनती है जिसके थ्रू pollen grains बाहर आते हैं. The stomium and septum dissociate. जो stomium और septum है dissociate होता है due to tension developed by dehydration in mechanical layer or due to chemical disruption brought about by hydrolytic enzymes. तो जो endothelium है वहाँ पे hygroscopic movements होते हैं जिसकी वजह से stomium और जो septum है वो dissociate हो जाते हैं. With the loss of water, the differentially thickened dead cells of endothelium contract from their outer thin walls and become concave. तो जैसे ही डिहाइड्रेट हो जाते हैं एंडोथीशियम के सेल तो बाहर की जो उनकी वॉल है, थिन वॉल, वो कॉन्ट्रैक्ट हो जाती है. दैट गिव्स एन अपीयरेंस ऑफ कनकेव that becomes concave and thus bringing their outer radial wall nearer, जिससे कि उसकी बाहर की outer radial wall नज़दीक आती है. As a result, the endothecium shortens, जिससे जो endothecium है वो short हो जाती है, and ruptures the anther lobe, और उसके result से जो anther lobe है वो rupture हो जाता है in the wall region of stomium and disperse the pollen grains. और स्टोमियम के रीजन पे ये जो सेल्स हैं वो ब्रेक हो जाते हैं, जिससे पोलन ग्रेन्स बाहर निकल आते हैं. So this is how antherdescence occur. This is the original photograph under microscope. So these cells are known as stomium cells, और जो एंडोथीशियम के सेल्स हैं वो रिस्पॉन्सिबल होते हैं पोलन ग्रेन की डेहिसेंस के लिए. Next comes pollen structure. So basically a pollen grain consists of two parts. One is the central cell structure, which consists of the cytoplasmic portion. It contains two cells. One is generative cell, the smaller one, and the larger one is the vegetative cell. So it consists of two distinct parts. One is the central cytoplasmic part, and second consisting of the wall of grain. जो pollen grain की wall है, that forms the second part. First is this central portion, and this wall shows differentiation. It consists of inner intine, also known as endospore, and outer exine. It is also known as exospore. The texture of pollen wall shows great variation. जो exine है उसमें बहुत सारी variations होती हैं, which varies from smooth to spinous, verrucate, baculate, and this is very important for the taxonomic point of views. It helps in classification of plants. These pollen exine patterns helps in classification. They have great taxonomic importance. Pollen grains of various plant species can vary quite a lot in size and aspect. That is round, oval, disc-shaped, bean-shaped, filamentous, etc. And they play a very important role in classification. They are mostly white, cream, yellow, or orange in color. If we talk about color, so they show color diversity also. Next, first is the central part. It contains cytoplasm, vegetative nucleus, generative cell, dictyosome, mitochondria, and endoplasmic reticulum. So basically जो pollen grain है उससे हम दो parts में divide कर सकते हैं. First one is the central part. So this central part consists of generative nucleus or generative cell or vegetative cell. In addition to this, it contains dictyosome, mitochondria, and endoplasmic reticulum. If we talk about its constituents, so mature pollen grains contain protein 7 to 26%, carbohydrate 24 to 48%, fats 0.9 to 14.5%, ash 0.9 to 5.4%, and water, that is 7 to 16%. So these are the various chemical constituents or constituents of mature pollen grain. The central cell contains vegetative cell. So it is the large size cell which is formed after asymmetric nuclear division. So it constitutes the major part of microspore after asymmetric division. Asymmetrical division means ये equal division नहीं है, unequal division है, so it occupies the maximum portion of pollen grain. It is rich in active dictyosomes, mitochondria, endoplasmic reticulum, free ribosomes, blasters, starch grain, RNA, and proteins. So ये सारी चीज़ें इसमें present होती हैं. This cell store reserve food that are utilized for pollen tube development and transport of male gametes towards the egg in embryo sac. So जो reserve food material है वो यूटिलाइज होता है पोलन ट्यूब फॉर्मेशन में, और मूवमेंट जो मेल गैमेट्स की होती है टुवर्ड्स द एग इन एम्ब्रियो सैक. सेकंड इज जनरेटिव सेल. सो दिस इज डेवलप्ड ऑन वन साइड ऑफ द पोलन ग्रेन. ये पोलन ग्रेन की एक साइड में डेवलप होता है. लेटर इट डिटैचेस फ्रॉम द वॉल एंड मूव्स इनसाइड द वेजिटेटिव सेल. सो ये वेजिटेटिव सेल के अंदर ही मूव करता है. द वॉल ऑफ जनरेटिव सेल थिन्स डाउन इन वेजिटेटिव सेल्स एंड इट बिकम्स स्पिंडल शेप. It is enclosed by two plasmamembranes, one of its own, एक इसकी अपनी होती है, and the other formed from detached invagination of plasmamembrane of vegetative cell. तो दूसरी इसमें होती है इन्वेजिनेशन ऑफ प्लाज्मा मेम्ब्रेन ऑफ वेजिटेटिव सेल्स की दो मेम्ब्रेन सराउंड करती है जनरेटिव सेल को. इट्स साइटोप्लाज्म कंटेन डिक्टियोसोम, माइटोकॉन्ड्रिया, एंडोप्लाज्मिक रेटिकुलम, फ्री राइबोसोम एंड माइक्रोट्यूब्यूल्स. सो ये सारी चीज़ें इसमें प्रेजेंट होती हैं. प्लास्टिड्स आर एब्सेंट एक्सेप्ट सिर्फ एक जीनस है जिसमें प्रेजेंट होते हैं प्लास्टिड, दैट इज ओइनथेरा हुक्री. सो इसमें प्लास्टिड्स प्रेजेंट होते हैं, अदरवाइज प्लास्टिड्स आर एब्सेंट इन केस ऑफ जनरेटिव सेल or microspore. Next comes male germ unit. It refers to the assemblage of this vegetative nucleus and two sperm cells. जो ये दो sperm cells हैं, ये तीनों चीज़ें as a single unit transfer होती है. So this assemblage is known as male germ unit. So, और इसका जो benefit है, it helps in synchronization of formation of endosperm as well as the diploid zygote. So it refers to a functional assemblage between the vegetative nucleus and two sperm cells in a flowering plant. So जो angiosperms हैं उनमें ये assemblage है vegetative cell की और साथ में दो sperm cells की, और as a unit ये transfer होते हैं through pollen tube, and this is known as male germ unit. So transported as single reproductive unit within the pollen tube and later delivered to the embryo sac to bring about the act of double fertilization. So और उसके बाद दोनों ही जो sperm cells हैं वो embryo sac के अंदर transfer होते हैं. They bring the act of double fertilization which involves fusion of one male gamete male sperm cells with egg to form diploid zygote, and second with the polar nuclei to form triploid endosperm. The significance of this reproductive assemblage ensures synchrony of the double fusion that subsequently resulted in the coordinated development of resulting zygote and endosperm. So इसका जो benefit है, significance है, वो है synchronized formation of this endosperm as well as zygote. The mechanism that accounts for the movement of sperm cells involved an ectomyosin like motility system as that is present in animal cells. So एक ectomyosin like motility system present होता है उसी की तरह जैसा कि animals में है. So that mechanism accounts for the movement of sperm cells. In plumbago जो दो sperm cells हैं उनका size same नहीं होता है, they are unequal sized. That shows preferential fusion with either egg or central cell. So size different है plumbago में. दो sperm cells का preferential fusion show करता है. It means कि एक उसमें से fuse करेगा egg के साथ, दूसरा central cell के साथ on the basis of their size. So this is what is male germ unit. Next comes pollen wall. So pollen wall consists of two layers. अगर हम pollen wall के structure की बात करते हैं, it consists of two layers. One is the outer layer that is known as axine. Second one is the inner layer which is known as intine or also known as endospore. If we talk about this intine, it is made up of pectocellulosic substances. और ये भी further दो layers में differentiate होती, that is act intine and end intine. While this axine is a very complex layer, or it is broadly divided into two layers again, that is act axine and end axine. So it is further differentiated into this tectum columella foot layer that we will discuss one by one during the pollen wall development. So it comprises of the outer layer called axine and the inner layer intine. और ये जो terms हैं pollen wall layers के लिए, these terms were given by Frisché in 1837. The intine is predominantly made up of pectocellulosic material. जो pectocellulosic substances है उससे intine बनी होती है inner layer, whereas the axine is structurally more complex and can be further divided into sublayers like we have sexine, that is act axine, and the knee axine that is also known as the end axine. So ये दो layers से बनी होती है axine. The axine is composed of sporopollen materials. Sporopollen material हमने पढ़ा है कैसे ये बनता है. In turn, this is formed by oxidative polymerization of carotenoids. This layer is acetolysis resistant means इसमें acetylosis नहीं होता है and can withstand both physical and biological degradation, so it provide resistance to the pollen grains. A specific number of germ pores or aperture occur in pollen wall. So specific number of pores जिसके through pollen tube बाहर निकलती है वो present होता है particular species में. So ये भी एक criteria है which is very important in the classification of plants, especially angiosperms. Next comes pollen wall development. So for this, tapetum plays a key role. As we know, it supports the development of microspore with enzymes and provide nutrients, metabolites, and sporopollen precursor and the pollen coat that is known as exine. So ये सारे काम करती है tapetum. Depends heavily on the tapetal layers of the anther. This pollen wall development depends upon this tapetal layer. As we know that these tapetal tissues are rich in dictyosomes, endoplasmic reticulum, and other nutrients. It helps in exine formation. There are two phases in wall formation. दो phases होते हैं pollen wall formation में. The first one involves wall material जिसमें स्पोरोपोलिन का प्रिकर्सर आता है आर कॉन्ट्रीब्यूटेड ओनली बाय द स्पोर साइटोप्लाज्म. जो स्पोर साइटोप्लाज्म है उसी से बनता है ये. दिस इज द फर्स्ट टाइप टू द पोलन टेट्राड स्टेज, और ये होता है अप टू द पोलन टेट्राड स्टेज इन द फर्स्ट फेज, while द सेकेंड फेज बिगिंस आफ्टर द सेपरेशन ऑफ माइक्रोस्पोर्स फ्रॉम द पोलन टेट्राड. जब माइक्रोस्पोर सेपरेट होते हैं पोलन टेट्राड से एंड द वॉल मटेरियल हेयर आर कॉन्ट्रीब्यूटेड बोथ by tapetal cells as well as by the spore cytoplasm. तो दोनों ही चीज़ें इन्वॉल्व होती है इसमें. That is tapetal cells as well as the spore cytoplasm, which helps in formation of the pollen wall. Next, why we need this exine? As we know that pollen grain contains very sensitive material, और इसकी प्रोटेक्शन बहुत ज़रूरी है. So that's why it contains this exine which contains sporopollen, that is the hardest material which protects the pollen grain. As we know that separation of microspore is brought about by degradation of callose wall, और जो callose wall है उसकी degradation करता है enzyme that is known as callase. ये भी tapetum ही secrete करता है. This is secreted by dictysome of the tapetal cell. Then rough endoplasmic reticulum along with its associated vesicles in the tapetum are responsible for forming sporopollen-like bodies in case of amoeboid tapetum, that is plasmodial tapetum, and sporopollen precursor is formed in the secretory, that is also known as parietal tapetum. So दो तरह की चीज़ें बनती हैं. One is sporopollen-like bodies which are formed by amoeboid or plasmodial tapetum, while sporopollen precursors which are formed by the secretory or parietal tapetum. And this sporopollen is this material is considered one of the nature's most advanced polymer and formed from oxidative polymerization of carotenoids. तो जो carotenoids हैं उनकी oxidation से ये बनता है. That is sporopollen, and it is one of the important or toughest material which protects the pollen grains. This has been formed to ensure that the tender cells inside the pollen survive from the environmental injury. So ये ensure करता है कि जो pollen grain के अंदर delicate or soft or sensitive cells present है वो survive करे from the external environmental injury or environmental stresses. Then pollen wall layer consists of exine and intine. Firstly, we will discuss about exine. As we know that it is the outermost resistant layer, which is a tough layer, and is chiefly composed of inert material which is known as sporopollen. बाहर से इसमें जो कोटिंग होती है sporopollen की होती है, and that sporopollen is toughest material which is an inert material जो कि prevent करता है pollen grain की degradation को. Then the outermost part of pollen wall shows various sculptured patterns and are made up of rarely oriented rod-shaped structure जिन्हें कहते हैं vacuolae. So बाहर से this is the exine. This portion is exine, which is further divided into two parts. That is sexine, also known as ectexine, and next one is the neoexine, that is also known as endexine. So जो बाहर वाला portion है, it consists of rod-like structure जिन्हें कहते हैं baculum, that is plural, baculum is singular, and they also have flat plate-like structure जो कि baculae के ऊपर present होता है, जिसे कहते हैं tectum. This is called tectum. So the baculum may be free and enlarged at the top, और may be fused at the top to form a flat layer known as tectum. So ये free भी हो सकते हैं और ऊपर से fuse हो सकते हैं to form a flat plate-like structure जिसे कहते हैं tectum. The tectate walls have various types of pores and opening on to the outside. तो इसके ऊपर कुछ pores और opening present हो सकती हैं. Different sculpturing patterns of exine have been shown to have great taxonomic value that I have already discussed. जो exine के patterns हैं, different patterns sculpturing, they helps in classification of the plants, so they have great taxonomic value. The exinopollen grain of insect pollinated plants is coated with a sticky substance that is known as pollen kit and trephine. तो ये बहुत ही important एक येलो कलर का सब्सटेंस है जिसे कहते हैं pollen kit और trephine, that we can see in this photograph. जो बाहर का ये yellowish color का fatty substance है, that is known as pollen kit, and it helps in sticking to the insect during pollination, और ये attract करता है insects को pollination के लिए भी. This is a hydrophobic substance. It is formed by breakdown of elaioplast and tapitosome at the late stage of pollen development. तो ये दो चीज़ें responsible है, जो elaioplast है which store fat and tapitosome, इनके breakdown से बनता है pollen kit at the later stage of pollen development. Then this pollen kit is a hydrophobic substance and attract the potential insect pollinators, और ये attract करता है insects को for pollination purpose. While this trephine is a hydrophilic product and it is formed from degenerating remains of tapetum. So tapetum से ही बनता है ये भी, but this is a hydrophilic substance. This photograph shows outer layer, that is exine, and inner intine. Intine is composed of the pectocellulosic substances, cellulose, hemicellulose, callose. Next layer is intine, which is the inner layer of pollen wall. This is inner and smooth layer of pollen wall and is mainly made up of pectocellulosic substance. This is the intine. This layer is usually thicker and has plates of enzymatic proteins in the vicinity of germ pore or aperture. This layer is chiefly composed of polysaccharide, that is pectocellulose, a polymer of cellulose and fructose. Next, pollen aperture. It is a aperture that helps in growth of pollen tube. जो pollen tube है वो इन apertures के through बाहर आती है. So these are known as apertures. It serves as an exit for pollen content, becomes spore or furrows, also known as colpi, present in majority of eudicots, और जो true dicots हैं उनमें present होता है ये. Mostly जो pollen grains होते हैं they are tricolpate. It means they have 3 furrows or 3 pores through which the pollen tube can come out of these spores. The arrangement and number of pores and furrows are chief criteria for identifying pollen. तो ये भी एक identification marker है that how many apertures are there in the pollen grain. So we use the term colpus, and we have different patterns of these apertures and furrows which helps in identification of pollen grains. We have one system that is named as NPC system for pollen classification. The N stands for number, P stands for position, and C stands for character. This is a vast topic. I will not go into detail of this topic. We will discuss it briefly. This NPC system, N stands for number, P stands for position, C stands for character, system of pollen classification was given by Erdmann in 1969. There are 9 classes of pollen grains on the basis of number of aperture. अगर हम number की बात करें तो उसके basis पे हम 9 classes में divide कर सकते हैं pollen grains को. And we use the term treme for aperture, and pollen without aperture are known as or called as atreme, that is designated as N0. While monotreme, it means there is single aperture indicated by N1. Then ditreme, it means having 2 apertures. Then polytreme having many apertures. Pollen with irregular apertures are called as anomotrim. जिसमें irregular aperture present होते हैं उन्हें कहते हैं anomotrim pollen grains. Then we have 7 classes on the basis of position of apertures and 7 classes on the basis of characters of apertures. I will not go to the detail of these classifications. So this is a brief idea of NPC, system of pollen classification. Next comes pollen viability classes given by Harrington in 1970. ये जो classification it is based on the viability of pollens for a particular duration. Firstly, we have long-lived pollen grains. So these pollen grains are viable for 6 months to a year. तो ये 6 months से लेके 1 year तक viable रहते हैं. We have examples like we have some families: Ericaceae, Saxifragaceae, Rosaceae, Fabaceae, Anacardiaceae, Whiteaceae, and Primulaceae. So in families के जो pollen grains है, they remains viable for a duration of 6 months to a year. Next, we have pollen with medium lifespan. These are viable for approximately 1 to 3 months. तो ये 1 से लेके 3 month तक viable रहते हैं. Examples are the pollen grains of family Liliaceae, Amaryllidaceae, Salicaceae, Ranunculaceae, Brassicaceae, Rutaceae, Sacrofulariaceae, and Solanaceae. The next comes short-lived pollen grains. As the name indicate, इनकी जो viability है वो बहुत कम समय के लिए होती है. They are viable for few minutes to a couple of days. We have examples of families like Alismataceae, Poaceae, Cyperaceae, Comalianaceae, and Juncaceae. So ये कुछ families है जिनके pollen grains बहुत ही कम समय के लिए viable रहते हैं. So this was classification on the basis of pollen viability and was given by Harrington in 1970. Next comes male gametophyte. As we know that microspore represents the first cell of gametophytic generation. जो microspore है वो represent करता है first cell of gametophytic generation, that is the haploid generation. It is densely cytoplasmic with a centrally located nucleus. जो microspore है उसमें dense cytoplasm होता है and centrally located nucleus. Due to increase in size of vacuole, microspore nucleus is pushed towards the pollen wall. तो center में एक vacuole appear होता है जो increase होता है size में, that pushes the nucleus towards the pollen wall. The microspore nucleus may undergo period of rest before starting to develop into a male gametophyte. ये microspore का जो nucleus है, it will not immediately divide, it can undergo a period of rest, and after that it will start developing into a male gametophyte. This rest of microspore nucleus may be for several weeks and to a month, or it can be up to an entire winter, as in case of Betula odorata. So जो Betula odorata है इसमें जो pollen grains का microspore का nucleus है, that remains dormant during the winter period, entire winter. In plants growing in temperate areas, जो कि grow करता है temperate region में जहाँ पे snowfall होता है, while it starts dividing immediately, that is less than 4 days in Tradescantia reflexa. जबकि ये डिवाइड करता है immediately, that is less than 4 days in Tradescantia reflexa, जो है plant tropical region का, that is of warmer region. Next, the microspore nucleus divides through mitotic division. ये डिवाइड करता है mitotic division से, and the pollen mitosis is asymmetric. और ये है asymmetric, it means कि 2 equal halves में cells divide नहीं करते हैं. It divides asymmetrically. The resultant nucleus towards the center is called vegetative nucleus. Center की तरफ जो nucleus बनता है उसे कहते हैं vegetative nucleus, and the one near the wall is known as generative nucleus. जो wall के पास है उसे कहते हैं generative nucleus. The phragmoplasts form a more or less curved watch glass shaped ball between the daughter nuclei, leading to formation of 2 unequal cells. तो phragmoplast एक curved watch glass की shape की wall बनाते हैं इन 2 nuclei के बीच में, and results in the formation of 2 unequal cells. This new wall does not fuse with pollen wall at the ends, but forms a lining to the mother cell wall that cuts off generative cells from the vegetative cell. तो इसके रिजल्ट से हमारे पास बनते हैं 2 cell. One is the vegetative cell and second one is the generative cell. So this division results in formation of 2 unequal cell. One is the smaller cell, that is generative cell, which is formed away from the aperture and is known as generative cell. It will ultimately give rise to 2 male gametes. ये बनाता है 2 male gametes. The larger vegetative cell comprising most of the cell, ये most maximum part occupy करता है cell का and will give rise to the pollen tube in future, और ये बनाएगा future की pollen tube. Later the generative cell detaches from the pollen wall and it moves to lie inside the vegetative cell of the cytoplasm. Vegetative cell cytoplasm. तो ये जो vegetative cell है ये pollen की wall से detach हो जाता है और lie करता है inside this cytoplasm of vegetative cell, and this is a very unique phenomenon where one cell comes to lie inside the another cell in case of angiosperm. So this is a very unique phenomenon in angiosperm. The generative cell finally becomes oval or spindle shape and pollen are shed at this stage in most of the angiosperm. और angiosperm में जो pollen grains है वो इसी stage में release होते हैं, mostly 2-celled stage, and in some cases 3-celled stage में भी pollen grains release होते हैं. Next comes pollen allergies. So जो pollen grains है they are known to cause some serious allergies. Pollen grains can cause allergy. Pollen in the air enters the nose and throat where they can cause allergy and asthma symptoms, known as the most common one is the hay fever or allergic rhinitis in people who are allergic to them. So pollen grains करते हैं serious allergies cause. इसमें से most commonly जो allergy that is hay fever and asthma. So the common symptoms हैं allergy के, pollen allergy के, runny nose, nasal congestion, coughing, sneezing, watery eyes, itchy nose and eyes, then worsened asthma symptoms. These pollen grains can lead to serious asthmatic problem and decreased quality of sleep. So जो नींद की quality है वो भी reduce हो जाती है. So we have lot of plants which produces pollen grains that can cause allergies. So we have some preventive measures like overall avoidance, pollen grains से बचना है, then check pollen counts daily and stay inside the high pollen days. जो pollen grains ज़्यादा release होते हैं तो उस समय हमें अपने आप को protect करना है pollen grains से. Then wash bedding and clothing often in hot water. Shower and change clothes after you have been outside on high pollen day. तो proper अपनी cleansing करनी है to avoid the pollen allergies. Next comes abnormal characteristics in pollen grains. So there are some facts about abnormal characteristics in pollens. Firstly, we have some marine plants like Halodule, Halophila, Thalassia, Zostera, etc. They have finger-like pollen grains or thread-like pollen grains which are known as filiform pollen grains. They can be up to 5 mm long and without well-defined exine. इसमें कोई clear-cut exine present नहीं होती है. Then there are some families जिसमें कि microsporogenesis के result से जो microspores बनते हैं, only one microspore is functional. So only one of the daughter nuclei develops into functional microspore after microsporogenesis. So जो microsporogenesis का event है उसमें सिर्फ एक single nuclei ही functional रहता है in Cyperaceae, like we have members of Cyperaceae Cyperus and Kalinga Serpus. The rest 3 non-functional nuclei are pushed to one side of the microspore mother cell where they are separated from the functional nucleus. Then pollen embryo sac-like structures were first reported in Hyacinthus orientalis. एक प्लांट है Hyacinthus orientalis, उसमें pollen embryo sac की तरह स्ट्रक्चर देखे गए हैं. This phenomenon was observed by Nemec in 1898. So Nemec ने सबसे पहले ये phenomenon को observe किया है. And occurrence of pollen embryo sac is also known as Nemec phenomenon. इसे Nemec phenomenon भी कहा जाता है. This is very unique phenomenon. So he believed that they are formed by 3 successive division of vegetative nucleus. तो Nemec ने ये observe किया, he had hypothesis that जो embryo sac बनता है, pollen embryo sac, ये बनता है 3 successive division of vegetative nucleus. But later on ये observe किया गया कि जो ये pollen embryo sac बनता है, it can be formed by 4 ways. Namely, first one is division of microspore mother cell. एक तो ये microspore mother cell के division से बन सकता है. दूसरा, it can be formed by microspore nucleus. Third, it is formed by vegetative nucleus, and fourth one by generative nucleus. So there are 4 ways which can give rise to this Nemec phenomenon, that is pollen embryo sac. और ये जो property है कुछ plants की to divide and give rise to this structure, that leads to a phenomenon which is known as एंड्रोजेनेसिस. सो एंड्रोजेनेसिस जो गुहा एंड महेश्वरी ने 1964 में टिश्यू कल्चर में काम किया है, that these anthers can give rise to the full-fledged plant that is known as androgenesis. So इसी phenomenon से उसमें help मिली है. So now it is reported to occur in many other plants. जो ये नेमिक phenomenon है, ये बहुत सारे plants में देखा गया है, and this is regarded as an anomalous feature. So this is an abnormal feature. So this was all about this microsporogenesis and other facts which were related to this topic. Hope you will get some idea from this presentation. If you have any questions, queries and any suggestions, you can give it in comment section. Thanks for watching. Have a great day.